Hi guys, Dr. Gillard here once again. Let's do some embryology, shall we? It is week 4. It's Monday. It's the spring of 2020 quarter, and off we go. So last time we left off, we created a morula, and that's a 16 for our purposes, 16 cell little creature here. Doesn't look much like a human, doesn't it? Still has a zona pellucida around it. Corona radiata's gone, about day 2, that one's gone. We're on day 4. And yeah, and I forgot to mention what sweeps. Remember we had the secondary oocyte here, and it goes down, gets fertilized right about here. And it goes down, right? And we're right about here with it right now. And it's these cells within it have been dividing and dividing. We have a 16-cell morula because it looks like a mulberry. But how does it move down here? I forgot to tell you, there's just like how fecal material moves through the intestine, there is a peristaltic contraction that goes on and helps push the egg down the fallopian tube. In addition to that, there's ciliated simple columnar cells that also live on the oviduct or the lumen of the fallopian tube. And the combination of those two is what's moving this down toward the uterus. That's where we're headed now. So we get to talk about segregation. So this is the first cell segregation occurs after the morula stage, about late in day 4. So the ball of blastomeres begins to segregate. Remember the blastomeres? They're called daughter cells in other cells in the body, but we call them blastomeres here. We're up to about 16. And they start to segregate. So that means we must have some gene populations turning on for the first time. But basically, we're going to shape an inner cell mass, a central population. So cells that are randomly kind of scattered out through our little ball, they're going to start to clump into an inner cell mass, and then some are going to kind of push to the outside to form the trophoblast. Some people, only actually one book I think, Langman, which is a board of chiropractic examiner books, calls it the outer cell mass. I've never seen that anywhere else. Langman likes to do that. It seems to like to make up words that I don't see used anywhere else. But anyway, so trophoblast. All right, and before a little mini rabbit hole, I assume you know this, the central dogma, right? I'm not going to go over this too deeply, but go to the Amoeba Sisters. There's tons of videos on the central dogma. But remember, DNA, the central dogma, you may or may not say central dogma. Well, here's the central dogma right here. It's DNA is replicated through DNA polymerase. We have to, when cells divide, when they go through interphase, they're replicated. And some say that's the start of the central dogma, where the DNA is replicated. Then the DNA is broken open and read by an RNA polymerase. The template strand is read, and that process is called transcription. RNA polymerase creates a single strand of RNA called messenger RNA, and that swims. This is happening in the nucleus. This swims out of the nucleus, and it hooks up with a ribosome, and the ribosome is able to read this transcript. That's called translation. And the transcript, the ribosome translates the coding here into usually amino acids, or usually some type of protein. Could be a growth factor, could be just a protein building block of, let's say, a transporter. How about like an aquaporin's transporter? It also makes RNA molecules too, could go through this process, a messenger RNA, transfer RNA. And yeah, so that's the central dogma. So I assume that you know that. When I say gene expression, when I say genes are turned on, that's really gene expression. And that's the same as saying that genes are turned on and running through the central dogma to express their ultimate protein or their ultimate product, which is whatever they happen to express. Okay? Also remember there's 23 pairs of chromosomes, 46 in total. These are, each one of these, here's a pair of homologous chromosomes. Let's say it's chromosome 9. And here's the parts, the centromere holds them together. But remember, there's coding regions within each one of these, and they're also kind of homologous. But those are called genes. And these genes contain the information that the RNA polymerase reads to make a transcript, to make an mRNA transcript. So genes live on chromosomes. Okay, pretty basic stuff. Notice there's a gene deletion here that causes all sorts of mutations. We could have mutations in these things. We'll talk a lot about mutations, but there's a deletion mutation. All right, so I think we got that pretty much covered. There are in a human cell, you name the cell, pull it out, and you can find all the chromosomes that pair up like this. One pair of sex chromosomes are weird. Is this a male or a female? This is a female. It's an XX. But those are, you could technically say that there's 22 pairs of, that shouldn't be genes, 22 pairs of chromosomes and 1 pair of sex chromosomes. I need to make a note and fix that. I don't even know what slide we're on here. Slide 171. Okay. All right. So genes can be turned on or turned off. If you turn off a gene, the RNA polymerase won't read it. It's closed for reading. If you turn on a gene, that means it's amenable to being read by the RNA polymerase, and you can make a transcript and get that transcript read by a ribosome and make that protein. So we'll talk about autoimmune diseases. Some autoimmune diseases occur when certain irritants like cigarette smoke particles and Berger's disease, for example, when they turn on certain genes that aren't supposed to be turned on, and they turn on so many in cells, whether they be Berger's disease, we're talking about endothelial cells of blood vessels, they turn on so many genes that all of a sudden the body's immune system, it doesn't know who they are anymore. It's like, who the heck are you guys? You look foreign to us, and it attacks, and you get an inflammation, an autoimmune type inflammation. We'll talk about that scenario quite a bit as you go. By the time you get in 5th quarter, we'll talk about that a lot. All right, so how does that first segregation occurred? Right, because remember, back before we fell in that little rabbit hole. Oh yeah, I did put the rabbit hole sign. Our little ball of cells all of a sudden is starting to kind of segregate. It's 2 different populations. We formed an inner cell mass and a trophoblast, but how exactly does that occur? Oops, went too far. All right, so certain genes within the inner cell mass, they wake up. And what wakes them up, we don't know for sure. There's ideas, but there's no conclusive evidence of what wakes them up. But they turn on. And once they turn on, the new RNA polymerases are able to read them, and we can start expressing gene products. What does that mean, expressing gene products? Well, RNA polymerase can read the gene because now it's open for business, right? And we can start making proteins. And these products, they tell these blastomeres to do 2 things. One, to segregate or move to the center, and then the other was to move to the periphery. And that's to form the inner cell mass there and the trophoblast around the outside. Yep, and so that's our earliest example of gene expression. So let's meet the inner cell mass and the trophoblast. So the inner cell mass is also called another weird one that's only seen in Langman. I don't see it in any other of the books, but chiropractors watch out, or chiropractic students watch out, because that's fair game for boards. It gives rise to a really important, this inner cell mass is basically us. This is the little human. And it gives rise to the 3 germ layers, which turn into the little human. Some authors say it gives rise to the intraembryonic tissue, or the embryo. The trophoblast is, again, weird outer cell mass by Langman, but some authors call it the extraembryonic tissue because that is not us. That's going to shelter us and feed us and protect us as we develop, but it's not going to become part of us. In fact, tropho that means nourishing. So it's kind of nourishing type of deal. We'll see as we go through. But it ends up forming the placenta, for example. Right? So now our little trophoblast is segregated. So we have cells in our little pizza here. We have 6 cells that have formed the inner cell mass. And now we have, I don't know how many, 6, 8, whatever, form the outer cell mass, which I shouldn't call, that's that weird AKA, but it forms the trophoblast. All right, so we got our first segregation. It doesn't look much like a human, does it? But the human will form out of this light blue stuff, and this will form the placenta here in a long time from now. So to become now, watch out for the little geeky guy here. This means it's going to get technical. You guys who want to crush the class with an A, you got to know the little geeky guy stuff to get the A. If you're just trying to pass the class and get through the program, I wouldn't worry too much about the little geeky. You could probably still get a C on the test without knowing the little geeky stuff. But anyway, to become inner cell mass cells, you specifically, what genes need to be turned on? I didn't tell you that, but let's tell you. So the very first 2 genes that are turned on are the Wk4 gene and the nanog gene. Wk4 and nanog. Right? And later we'll see this nanog gene turns on another gene which creates the epiblast and hypoblast. So we're not done talking about nanog. Right? So these 2 gene products, which are growth factors or transcription factors, they also turn around and turn on some more genes. So new gene products turn right. They don't make any proteins or building blocks from anything, but they make kind of messengers that wake up other genes. And who do they wake up? They turn on Wnt, WNT, call it Wnt, and nodal, a really important one. So both of these are woken up. So now we got 4 genes on in the inner cell mass cells. How exciting. Right now, how many genes do we have turned on? Hundreds, thousands of them are turned on in a cell. But right now we got 4 of them. What about if you want to become a trophoblast cell? How does that work? Well, to become a trophoblast cell, we can't have Wk4 or nanog. We can't have any of these functioning. So we need a suppressor gene. We need a gene that's turned on and doesn't let anything else, doesn't let those guys turn on. And so the genes that get turned on in trophoblast cells, that's the CDX2 and the GATA3. And those are the ones, they inhibit the expression of Wk4 and nanog. Okay? And yeah, if those Wk4 and nanog can't be expressed, or their gene products can't be expressed, they won't become inner cell mass cells. They have to become trophoblast cells. Got it? Let's do just a little bit of anatomy. You'll learn more of this in Gross 2. That's why I really think embryology ideally should be put in 2nd or even 3rd quarter. You would get a better understanding. Because this stuff you haven't even had yet. We haven't had Gross 2 anatomy. Nevertheless, they always stick it in 1st quarter. Medical school as well. I think just to get it out of the way. But I guess that's the foundation, the building blocks. It's the very start of all the structures you study. So anyway, this is the uterus. So this is the baby's going to grow inside here. This is an A to P view of the uterus. It's got 3 parts. It's got a fundus on the top. It's got a body. And it's got a cervix. The back side here, which is into the plane of Page, is where the little baby is going to attach, or the little morula, which is going to change names in a minute, but it's going to attach back there normally, and then it will develop inside this wall, and we'll look specifically at that in a minute. Right, there's the vagina right down here. That's where the sperm get injected into that, and they swim. There's an external os, a cervical canal, and an internal os, and they get up here, and then they go into the fallopian tubes, which are cut off. Right, histologically speaking, we should know a little bit about histology. I think Dr. Doe will probably go over some of this as well. But there are 3 layers that we should talk about. There's a perimetrium. That's all I'm going to say about that, because it's really not important for us. But that's the outside. It's thin. It's like a serous or visceral peritoneal layer. And that's all I'll say. That's the very outer layer. Then we have the myometrium. This is the thick muscular portion. Super vascularized. A lot of blood vessels in here. This does not shed during a woman's period or menstrual cycle either. And a lot of smooth muscle in here. The contraction of that smooth muscle will help eject the baby when it's time to come out. All right. Now the endometrium we will talk about a lot. There's the endometrium. It's broken into 2 layers. Now your Junqueira, which is the chiropractic board book, they just call that the basal layer and the functional layer. Other histology books, I like this better, call it the stratum basale and the stratum functionale. So those are the 2 layers of the endometrium. So our little egg would be floating around. Here's the uterus, the cavity of the uterus. Our little egg's going to be dropped in here in a second. It's got to bind to the epithelium through a couple of receptors, and then it gets sucked inside the endometrium, and it pulls itself in here and attaches to all these blood vessels, and it starts to grow and grow and grow, and it gets pretty big. But this is where growth will occur. There's a uterine gland right there. We don't need to talk about that. Epithelial cells we'll talk about those a little more, because that's what's going to bind our little morula. Right? Stratum basale, as I said, that's basal layer. Those are stem cells. So the stratum basale, which is right here, didn't put the basal cells in here, but there would be cells in here. And they're stem cells. During the woman's period, they lose this whole layer. Stratum functionale is lost, sheds, but this doesn't. How does this come back then? Because there's stem cells here, and almost like the skin, it repopulates this whole system. Quite amazing, actually. All right, so stratum basale has stem cells. Yep, it's not shed during menstruation. Keeps the deep part of the uterine gland. Its stroma or lamina propria is an AKA for... It's always confusing histology. What lamina propria is an AKA for stroma, is an AKA for interstitium. And yeah, that's what that is. Stratum functionale, or the functional layer, that's what's shed during menses. Has a surface epithelium, which is simple columnar. Has a simple columnar, and you're learning about that in histology, right? We know what simple columnar cells are, right? They're just little tall cells. Simple columnar. Simple cuboidal would be little cubes, but that's what they are. They sit on a basement membrane, right? And then they have a lamina propria underneath, or stroma, if you will. And yeah. That's also the interstitium. There's blood vessels in here and capillaries, and blood has to swim through this interstitium to feed these cells. The other weird thing about these cells, some of them have cilia on them, and some of them don't. Some of them are little juice secretors, right? So some are ciliated, some are little juice secretors. Okay, so some are ciliated, some are secretory, and they go crazy. Changes occur in this whole region cyclically. All right, so here's another kind of a coronal view, kind of sliced right through, and you can see the cervix here, and you can see the endometrium. You can't quite see the endothelial layer of the endometrium, but it's there. Myometrium. Some sperm have gotten in here trying to swim up, and yeah, so now we kind of know where we're going. So where are we at with our little story? So our little morula, fertilized morula, is hanging out right about here. So we need to get it to drop into here. And the trouble is there's a sphincter right here that's shut. So we have to open that sphincter up. And remember the corpus luteum? We talked about ovulation and how the follicle turned into a corpus luteum, which secretes progesterone. That progesterone is going to open that sphincter when the body progesterone levels get high enough. It's going to open that sphincter, and allow our little morula to go kerplunk. Way ahead of my slides, though. Here's just some histological stuff here. That's our blastocyst. That's where we're headed. There's the simple columnar. You can't see the cilia. I'm not sure. I don't think those are cilia. But yeah, I'm not going to test you on that. That's Dr. Doe's domain. All right, kerplunk. So about day 5, by now we're 36 cells. Morula started out 16. 36 by day 5, our 36-cell morula goes kerplunk, comes out of the fallopian tube and drops not into the martini glass, but it is a martini glass. It's filled with a uterine fluid. And yeah. So the hormone progesterone, as I just said, secreted from corpus luteum, relaxes the little sphincter that's right here to help it plunk in there. And then the morula's in fluid, in uterine fluid. And now we have a problem because our little morula ship it's not really sinking, but it's leaky like crazy. So we need, and how does it get leaky? So a new set of genes, and we won't go into the genes that are turned on, but I guess you could just call them the sodium, potassium, ATPase genes, because they're going to make these. You're going to hear about these genes in physiology over and over, and we'll talk about it in the 5th quarter. Sodium, potassium, ATPase pumps. And those guys will pop up on the zona pellucida and on the plasma membrane of the morula, and they'll start taking in sodium. And who follows sodium like a puppy dog in most cases, except from the ascending thick limb of Henle, which you learn? Water. Where sodium goes, water almost always follows. So we start taking water inside the morula, and it starts getting in between the blastomeres. Okay? And yeah, so it starts accumulating fluid, cavity forms, and now we get a new name. So this cavity is called the blastocoel or blastocyst cavity. So we still got a trophoblast. That was our one layer. Then we got our inner cell mass, which is now started to drift toward one side of the cell. And yeah, so this is going to turn into the primary yolk sac, which is going to turn into the secondary yolk sac, and we'll follow the progression of this thing. There's a process called cavitation that makes this occur, and we won't say anything more about that. But because we have a really new recognizable structure, we have a cavity within our little creature here, we give this a new name, and it's named after the blastocoel. So it's no longer called the morula at this point. It's called the blastocyst. And remember, this is day 5. We're not even implanted into the uterine wall yet. We're still in the martini fluid. We're in the uterine fluid in the martini glass, right? So meet the blastocyst. Blastocyst still has zona pellucida, and I'll tell you right now, corona radiata is pretty much gone by now. Day 2, it's gone. But with this corona radiata, we can't bind. So we need to, the next task is to get rid of that corona radiata. Beautiful image right here. Zona pellucida is kind of degraded, but you can still see the ring around here. Plasma membrane, sodium, potassium, ATPase pumps have opened up and allowed fluid to leak in, and the fluid carved out a new cavity called the blastocyst cavity. Okay, you can still see the trophoblasts. These are trophoblast cells, which are going to get a name change pretty soon, all of these. They're actually going to give rise to something really important. We'll see in a minute. All right, there's a cartoon of that. Day 5, a lot of stuff going on. All right, so the inner cell mass now by this cavity is actually pushed up to one side of the blastocyst. And so wherever this inner cell mass attaches, so it'll be right here, it's called the embryonic pole. It's almost like the North Pole and the South Pole. So this north pole, that's the embryonic pole, right directly 180 degrees away from it. That is called the AB embryonic pole, the abembryonic pole. Okay, so the inner cell mass, or there's that embryoblast again, it's always toward the embryonic pole. This is actually going to be the dorsal side of the little developing human, and this is going to be the ventral side as we go. All right, so embryonic pole is up here. AB embryonic pole is down there. All right, here's another just beautiful image. You can see this blastocoel. Blastocyst cavity is well developed. And our little blastocyst. Did I call it a trophoblast? I hope I didn't. It's a blastocyst out of the blastocoel. Trophoblast is still, I always think blast is going to give rise to something. That's why it's kind of a weird name. Maybe that's why Langman is trying to name it the outer cell mass, which, I mean, really, it makes more sense. Easier to remember for sure. Anyway, yeah, you can see everything nicely there. All right, now, as I said, the next job, we're not going to do anything. In order for our little human to keep growing, it's got to get inside the endometrium. And so far, we're floating around the fluid, and we can't bind with that darn zona pellucida. So it's time to free ourselves from the zona pellucida. So, late in day 5, the blastocyst literally hatches, and that's what they call it, blastocyst hatching, literally hatches from the zona pellucida. As I said, it can't implant until hatching occurs. And it's thought, they're not 100% sure because we don't have humans to study. Based on animals, it's thought that the inner cell mass releases some enzyme that dissolves the zona pellucida, because this hatching occurs over the inner cell mass. So after the hole is made by enzymes, here's a cool picture of it really happening. So after that hole at the embryonic pole is made, it kind of works its way out of there. We won't get into the mechanism because we don't know in humans for sure. But yeah, we're leaving the zona pellucida behind, and the naked blastocyst is shot out into space, into the Martini fluid. And now it is ready. It's got receptors exposed now that can bind to the endothelial cells. Right? The endometrium, what's going to happen to the zona pellucida? It just withers away, and even when people get pregnant, they still have a tiny little period. Kind of fakes them out sometime. But it'll come out with that. Okay, what happened to the corona radiata? We said corona radiata was lost way back on day 2. And where the morula, or I guess embryo, was still in the fallopian tube, or even the zygote at that point is probably a better word for it. Okay, that was the corona radiata. Remember from the previous lectures, the cellular loose cells up here. All right, so let's keep developing our little human. Yet we're not even implanted. But now we have an intercell mass that we can further differentiate. And that's what we're going to do. So the inner cell mass itself is going to split into 2 layers. It's going to split into an inner layer and an outer layer. The outer layer, close to the embryonic pole, it's going to take on, it's going to form like a columnar epithelial type format. And that's going to be called the epiblast. We're going to be talking about these terms for a long time, the epiblast and hypoblast. In fact, this epiblast right here, this guy deserves a circle here. You want the ultimate stem cell? All researchers just would give anything to have are these epiblast cells. These give rise, these are the father or mother. This is the primordial cell of everything in your body is derived from epiblast. It's going to convert into an ectoderm, but these epiblast cells give rise to the mesoderm, The endoderm, the 3 germ layers, is all made by this epiblast. So it's a super important cell. There's no other cell that can turn into anything. Turn into a heart cell, a liver cell, you name it, your eye cell, your heel, your tibia. It can turn into anything. It'd be great if we had these to experiment with. All right. And the other cell, which is more toward the AB embryonic pole, this is called, because it's low, it's called hypoblast. And some primitive endoderm has been used again. I think it was Larsen again. I've never seen that anywhere else, but Larsen likes to do that. All right. The also thing about the hypoblast, they're cuboidal, and these are columnar. All right, so let's draw our little, this is a nice little drawing here. And we can see here's our trophoblast. This is our blastocyst cavity or blastocoel. We have hypoblast layer is now quite visible. And now we have a nice epiblast layer surrounded by a trophoblast. So we haven't even implanted yet. We haven't talked about these little receptors, right? We got some receptors here just waiting to bind to the epithelial cells of the endometrium. That's where we're going here in a second. But we got to get back to the little nerdy guy, because I know some of you go, well, what genes are turned on to do this? How come these cells go down here and these cells here? Remember, this was just one unit. What causes them to differentiate? Well, fibroblast growth factor 4, FGF4 is the key here. So cells destined to cluster in the dorsal aspect of the inner cell mass toward the embryonic pole. In other words, these guys, ones that are going to become epiblast cells, there's a gene that gets turned on called fibroblast growth factor 4, FGF4. Actually turns out that it was induced. What's that mean, induced? By nanog. Nanog was one of the first genes turned on. Induced means it was stimulated and turned on by. That's what induced means. And I give you these are like my teaching notes, so I give you hints here. But in the test, I'm going to say induced. I'm not going to say induced, turned on by. I'm not going to help you. So make sure you know the more scientific terms. But it was turned on by nanog, and that's what did it. And then via mechanisms too complicated to get into, the gene product basically tells the cells to move toward the embryonic pole. And those cells with FGF4 become epiblast cells. So now you're wondering, well, what about the hypoblast cells? Do they have FGF4? No, they don't. So FGF4 in the new hypoblast cells actually soaks down. Let's go to here. So FGF4, let's see, should I use blue? Let's use green. So FGF4 is being secreted, but some of it is soaking into these cells right here, neighbor cells. And this is happening all over. But just in this picture, kind of finishing these off, FGF4, I mean, maybe it's happening here too. If this cell hasn't differentiated, it could turn this one pink and that one would go down here as well. But the FGF4 soaks in to these cells, and it says, you guys are not going to be turning on FGF4. Instead, it has them, a new gene turns on called GATA6. Okay, so FGF4 turns on GATA6 in the cells destined to become the hypoblast. And then they're instructed to migrate down to the bottom toward the AB embryonic pole, right next to the blastocoel. And that's where they live. And that's eventually going to become the endoderm. Okay, so FGF4 is expressed in epiblast cells. GATA6 is expressed in hypoblast cells. Okay, inner cell mass. This is how it really looks, because we have a big inner cell mass, and all of a sudden some cells, they start getting FGF4 turned on in them. Okay, and once that's turned on, the FGF4 soaks into adjacent cells like this one, turns on GATA6, and that tells that one to move down here. And even that one up here is GATA6 turned on. It's going to migrate all the way down here and form that hypoblast. Okay. Another picture. I do like my pictures. It's one of the reasons I have so many slides, right? I love my pictures. FGF4 is turned on here. Got a 6. Now they're all lined up and the migration is done. You'd think we're done. We're not done. So meet the bilaminar disc, though. We'll talk about bilaminar disc and trilaminar disc. This is the start of the bilaminar disc. Bilaminar disc, who are the two layers? Epiblast, epi means above, hypoblast means below. There's our bilaminar disc. These have to morph into germ layers, germ cells, which haven't happened yet, but we're working our way that way. Okay. We also know once the epiblast and the hypoblast form, we can, with electron microscope and super powerful magnification, we can tell which way's up and which way's down. So we know that these epiblast cells, that's the dorsum. That's going to become the back side of you. This is going to become the ventral side of you. So we have just established what is called the dorsal ventral axis of the body has just been established. How has it been established? Once we have segregated the inner cell mass into an epiblast and hypoblast. Okay. All right. I think everything I said. Epiblast, hypoblast, dorsal, ventral. All right. All right, now let's do some more weird segregation, some more gene turning on and offing. All right, so one more segregation story. That might, for now, there's going to be a lot of segregation stories. But for now, I want you to meet the AVE cells, the anterior visceral endoderm cells. So hypoblast cells that are positioned toward the cranial region of the little growing embryo, those are going to undergo a transformation and become some very important cells, signaling centers, called the anterior visceral endoderm cells, or the AVE cells. Once they form, then we know the cranial-caudal axis as well, although usually the primitive streak tells us that. What's the deal with them? So let me show you a picture of one. Did I not put a picture yet? Yeah, we don't have an in-between. But once the hypoblast in yellow here is formed, we get some new genes turned on here. And that's going to make a new region called the anterior visceral endoderm. And then once this forms, we can tell this is going to be the head of the little creature here, and this is going to be the tail of the little creature. Right, but let's get to that point now. Okay, what causes the transformation? It's the blocking of nodal. So to make an AVE cell, nodal has to be turned off. Remember, it was turned on when the inner cell mass was first developed. Now you got to turn it off. And it was turned on by Oct4. Remember the first two that were turned on, Oct4 and Nanog? They turned nodal on. Now we got to turn that off. And it's not easy to turn off nodal. We'll talk a lot about the nodal gene, the nodal gene product, but it's a chore to turn it off. So here's how it's believed it's turned off. They're called nodal antagonists. And the ringleader of the nodal antagonists is a gene called DICOF1 or DDK1. And that's the one that soaks in and turns off nodal in those cells. It soaks into the hypoblast, the cranial region of the hypoblast, and turns those things on. So DDK1 blocks the expression of Wnt, which in turn causes nodal to be turned off. So that's the official way if you really want to get technical. DDK1 doesn't directly turn off nodal, but it turns off the keeper of nodal. Wnt is the gene that keeps nodal turned on. So if you turn off Wnt, nodal automatically gets turned off. See, hence the geeky guy here, or nerdy alert. This is nerd stuff. Okay, more nodal blocking genes. So just in case maybe you have a mutation in the DDK1 gene and oh my God, your fetus is going to abort. No, it won't. There's some other genes that can also do it. Lefty 1, some gangster type genes, the gangsters. This is the thinker of the gangsters, Cerebrus 1. Cerebrus 1 and Lefty 1, the two gangsters, those can also turn off nodal wall by themselves. They can assist DDK1 as well. So to recap, we have 3 genes that turn off nodal, DDK1, Lefty 1, Cerebrus 1, or CER1. Okay, so they're all on in cells that are going to become the anterior visceral endoderm cells, right? And they all directly or indirectly, they suppress nodal. We good? All right, now we made it back to this picture, and yeah, so DDK1, all the nodal gang, Lefty, Cerebrus, they're all turned on here, nowhere else. So that makes this the powerful AVE. So who cares about this AVE? Why am I making such a big deal of this? Well, they are important because eventually they will secrete some stuff. It's going to soak into these cells to make sure the primitive streak starts in the right place. Primitive streak is going to start caudally and work its way cranially when we form that. That's our little spine in our brain starting right there. But we don't want it to start back here. And so one of the jobs of AVE cells is to make darn sure this primitive streak doesn't start growing in the wrong place. That would be a disaster. Hence the disaster here. So inhibition, or it inhibits the formation of the primitive streak in the cranial, which is the wrong end, as I just said. So horrible mutations can result if that streak grows. The fetus probably won't be viable, won't make it, and won't live. But horrible mutations occur, like this mutated pig here. I mean, fetus is not going to turn. It's just to drive my point home. All right, how do AVE cells inhibit the primitive streak? Well, I said they soak up. Who soaks up? Well, the AVE cells secrete the same nodal inhibiting genes as we met before. So the gangsters, Lefty 1, Cerebrus 1, and DDK1. Those guys are going to diffuse into the cranial epiblast, as I just showed you, and it's going to turn them off, so nodal can't be expressed there. Without nodal, you cannot form a primitive streak. That's another job of nodal, as we'll see. All right, everybody good with that? What else do the AVE? They certainly do a whole lot more, as we'll see as we go through this. Cranial AVE cells, the ones that are more toward the cranial region, that's going to, all sorts of genes are going to turn on there. And those gene products are going to help form the heart and great vessels, some of the heart formation. The ones that are more caudal in the AVE cells, they're going to form another important structure called the precaudal plate, which we will talk about. Who cares about this precaudal plate? Well, that is going to start to form the early forebrain and the head. So that's a really important structure as well. Okay. All right. So enough of that stuff. All this stuff we haven't even bound yet. We don't even know if our little creature is going to make it or not. He's got to get inside the endometrium if he's going to make it. So let's make that happen. Uterine preparation. So the uterus has to be prepared to bind our little blastocyst. Remember, we changed the name from morula to blastocyst because of the blastocele has developed, the blastocyst cavity developed. So the presence of the developing blastocyst in the uterine cavity stimulates a change within the secretory epithelial cells. Remember I said some of them have cilia, some of them are secretory. The secretory ones usually secrete a slippery mucus stuff called mucin 1. It secretes mucin, which binds to water to make mucus. And if that's slippery stuff is being secreted, you're not going to bind. It's too slippery to bind to. So you got to turn those off. Secretory cells are turned off by mechanisms that we won't talk about. Okay, so mucin 1 is a glycoprotein, protects the uterine lining, makes it slippery. Yeah, so with the gene off, our blastocysts got the green light. So what happens next? And now we're about day 6 to 7, about a week after fertilization. So mucin 1 off, the blastocysts can bind to the epithelial cells, specifically the ones that have receptors called trophins on them. And more specifically, the trophoblast has receptors called L-selectins. So L-selectins bind with trophins to hook our little creature up with the endometrium. And now we have the first stage of implantation. And that's the goal, is to get our little fertilized embryo implanted. Or I guess we could call it a blastocyst implanted at this point. Right, so let's look at a cartoon of that. So the Martini fluid, there's our blastocyst. Blastocyst cavity is here. This would be our inner cell mass, but it's already segregated into an epiblast and a hypoblast, North Pole. See, this time the North Pole is down here, but the North Pole, the embryonic pole, is always where the inner cell mass is. And here's an L-selectin, and it's hooked up with a trophin, and we are off to the races. Epithelial cells now are, some changes are going to happen. Now, if you have a mutation in L-selectin or a mutation in trophinins, you're sterile. You can't have a baby. We fertilize just fine, but you can't. You have to be able to bind in order to get our little growing embryo implanted in here into the endometrium. All right, ectopic pregnancy. I thought we talked about this already, but if not, implantation usually occurs on the posterior wall of the uterine cavities I showed you at the start of the video. However, implantation can also occur in other regions where it's not supposed to. When we look, I swear we talked about this before, but I'll have to go back and check. Actually, let me make a note of that. 217. Do I really need this slide in here again? But anyway, so it can bind in weird places of the uterus. The fallopian tubes, a tubal pregnancy is terrible. Could even bind outside in the peritoneal cavity somewhere. Pouch of Douglas, maybe. So these are called ectopic pregnancy. They can not only destroy the baby, of course, they can kill the mom, because the mom can bleed like crazy. So I've actually had this on a couple tests before. So normally the ampulla is where fertilization occurs. It's not where implantation occurs. So if implantation occurs here, which is the most common ectopic pregnancy, tubal pregnancy, but specifically it's an ampullary ectopic pregnancy. That's the number one. In the fibula, in the infundibular region, still a tubal pregnancy. In the isthmus, still a tubal pregnancy. Down here in the cervix isn't good. It's pretty rare. But you could get one where the sperm come right out and fertilize the ovary or fertilize the secondary oocyte right when it comes out, and it binds immediately. Really rare. Also, ovarian ectopic pregnancy. And they didn't... here's the other ones. There's the pouch of Douglas here. The recto-uterine pouch, alias, about 1%. I guess it's not as rare as I thought. This is where fluid collects, so this is where, because of gravity, this is where it would go. And then it could even be on the intestines, but those are pretty rare. But I would definitely know those. I can see the question. All of the following are ectopic pregnancies except blank. And I could throw some weird word in there. So definitely know at least the 3 most common. All right, preparation. Okay, time to meet the crazy word here. This is a mouthful. Syncytiotrophoblast. Almost weird type cell population. So implantation stimulates our outer layer of cells called the trophoblast. It stimulates that to proliferate through mitosis. It spits out a new cell type. Spits it out toward the myometrium, because we're in the endometrium now. We're just penetrated the endometrial lining. So this new tissue is called syncytiotrophoblast, and it is weird. It has no each one of its cells doesn't have a cell membrane, yet it has a nucleus and mitochondria. And it has like a group plasma membrane around it. So maybe 4, 5, 6, 10, 12, 15 of these will share a plasma membrane. So a really weird cell, and that's a syncytiotrophoblast. So many nuclei in here. They've lost their cell membrane except for like one collective one. They're super aggressive. They're almost like a cancer. They have a leading tip that will see that secretes some enzymes, and it pushes its way through the endothelial cells as it moves deeper and deeper into the endometrium. And yeah, it's a really weird cell type. How does it, let's take a look at it before. So here's the very beginning. So here's our cytotrophoblast layer. Right? There's our, which one layer is this? Well, let's see. There's the inner cell mass. And the inner cell mass always points to the North Pole. So this is the embryonic pole here. So this must be the epiblast, and this is the hypoblast layer. But look what's happened to these cytotrophoblasts here. They're starting to divide, and they give rise to these weird things. So here's a cluster of 4 cells with no nucleus, or with no cell membrane, yet they have 4 nuclei. That's the start of the syncytiotrophoblast. And this is going to grow this way and grow and grow and grow, and it's going to pull our little embryo deeper and deeper and deeper into the endometrium. How does it cut through these cells and the other cells of the stroma it's got to get through? But the endothelial line is the one it's really got to get through. It secretes something called MMPs, matrix, what's that stand for? Matrix metalloproteases. And they can cut right between these cells. It's not going to destroy the cells. It's going to cut right between them. And that is how it goes. So it secretes matrix metalloproteases, or MMPs. They cut right through. I shouldn't say cut right through. It cuts between the cells. Let me make a note to fix that one too. It cuts between the cells. Yeah, and that's how it gets in there. Okay, and then as I said, it pulls the blastocyst deeper and deeper as it goes. The syncytiotrophoblast also gets bigger and bigger. It proliferates like crazy. And yeah, and that's the story with that. It's in the blood supply already. It's not just getting closer, but it's getting closer to the big blood supply of the deeper layer of the endometrium, which has some big vessels. But now it needs to hook up to those. We'll talk about how it hooks up to those vessels next time. But yeah, so there's our syncytiotrophoblast. It's pulled the embryo almost completely into the stroma. There's our cuboidal epithelial cells of the endometrial lining. It's not quite there, but look how big this syncytiotrophoblast is getting now. And it's starting to develop these little lacunae here, which are going to hook up to mom's blood vessels, as we'll see in the next lecture. And we're going to already get like a primitive circulatory system going here for our little creature. Okay. A little side note about those syncytiotrophablasts. They secrete something that is detected with pregnancy tests. They secrete human chorionic gonadotropin, or HCG. So the maturing syncytiotrophoblast starts cranking out a hormone. Hormones, remember, they get into the bloodstream and can travel throughout your body and cause all sorts of things. But they can be used, they get into the urine, and you can detect them and you can tell if you're pregnant. Normal people don't have this around. Right? So hormones signaling molecules, everything I said. And that's what's detected. Pregnancy tests, one way to do it. What's the job? Another job. So it's not just made for pregnancy tests, of course. It has a really important job. Let's go back to the follicle that's ruptured. And remember, that's by a week later, what's happened to it? Well, it should be just about dead, but it's not. Because HCG also binds to receptors on that dying corpus luteum, which is secreting progesterone. Remember we said progesterone prepares, it opens up the fallopian tubes, and it helps do other things. It prepares. There's some estrogens too that do things. But our corpus luteum is dying, but not anymore. The HCG mechanism is too complicated to talk about. It binds to the corpus luteum and it keeps it alive. It kind of stimulates it back into life. And for about 12 weeks, it keeps that corpus luteum cranking out progesterone and estrogens, which we need to develop the endometrium. So, yeah, it rescues the corpus luteum from early destruction. So HCG is super, super important. And remember, we're not going to go through this. We already went through this in a video. I just put that ovarian cycle in there. Genesis. That was a new video. I just put that in this quarter, so I don't need to go through this again. But there's our secondary oocyte. Ovulations occurred. And literally, this thing would be dead if it wasn't for HCG binding to this and keeping it alive. See, that one died. But it's not now because HCG is keeping it alive. Now we got a little house cleaning to do. So meet the cytotrophoblast layer. And it's just a name change, and it's really annoying. So once the blastocyst gets pulled all the way into the stroma, we get a name change. So now it's inside the endometrium. Right? It's all the way. It's not in the martini glass anymore. No more martini fluid for it. So once it gets in there, the trophoblast layer, we're not going to call it trophoblast. We're going to change the name and call it cytotrophoblast. Right? So it's cytotrophoblast cells. They were trophoblast cells. Same thing. I go in a big rant. Authors seem to forget this, and they keep going back and forth. You ever try to read an embryology book? You'll never figure this story out by reading any of these books. You have to read like 3 or 4 of them, and then you can get the story. And then you even have to go into PubMed and pull out some papers where the researchers, they're pretty good at explaining stuff. But anyway, it's just a name change. In the board books, all the books, Carlson's pretty good about it. But the other books kind of flip back and forth, and they forget that there's a name change, and it drives you crazy. But anyway, that's what's supposed to happen. Right? So once this guy is, actually could even be in a little bit further, but because this one's not completely in yet. This is the martini fluid, the uterine fluid out here. So it's almost in, but not quite. But once it's completely in, you get the name change to cytotrophoblast, and you say goodbye to trophoblast. All right, another problem. So think about this. Well, here's all sorts of blood, right? Blood vessel. Who patrols the bloodstream? White blood cells. White blood cells are going to see this monster, and what in the world's going to happen? They don't know who this is. They're going to attack this thing and would destroy it if it weren't for a protective mechanism. So once the blastocyst enters the endometrial stroma, mom's immune system spies it. And it wants to kill it, but it can't. So why doesn't it attack? Why doesn't it get destroyed by mom's immune system? There's something called EPF that's secreted from the blastocyst. So there's some proteins, there's some genes that are turned on when it starts to implant, and they release like a cloud around it, an early pregnancy factor. And that's what it's thought to act as a local immune suppressant. So for some, it's kind of like got a passport, or it's got papers, and it shows the papers to the immune system. Nope, we're okay. We got a pass. Stay away from us. And so it doesn't get attacked because of this early pregnancy factor. Okay, so that's the story with that. One more thing and we'll call it a day. So now we're on day 8. We're pretty much implanted. And the syncytiotrophoblast is invasion is going nicely. Started the 2nd week. And now we have a new cavity, a really important cavity that's going to form between the cytotrophoblast and the epiblast layer. And this is called the amniotic cavity. It's actually really important. So here, a little cavity starting to form right here. See it? And that's the amniotic cavity. That eventually is going to become huge and completely encircle this thing. but not now. It starts out just by a little cavity between cytotrophoblast cells. Remember, these used to be trophoblasts. Now they're cytotrophoblasts, and the epiblast layer. So amniotic cavity is right there. The actual size of this whole thing is that pencil dot. This is really, really small. It's just blowing up like crazy. So the amniotic cavity will eventually surround the entire embryo. It acts as a nourishing and protecting function, as we'll see. And yeah, we don't know too much about it because we're not allowed to study fetuses or aborted fetuses. So the ambion's an important layer. So immediately after this little cavity forms, our epiblast cells start cranking out a new cell line, and by mitosis, and these new cells go up to form a lining around the amniotic cavity. And so what a simple name. They're called amnion cells. One author calls them amnioblasts. Bauer, not a great book, I would add. Singh, not a great book for us either. Although they all have their places. They've always helped. All of these books have helped me piece together by reading them. I'm getting pieces of the story from all these different books. But these, by themselves, they're not nearly enough to make the story. Carlson, #1 book to buy is Carlson. Let's see. And I have notes, so I don't even know who he is, or just pure merit. So the new layer called the amnion. Okay, it's one of the 4 extraembryonic membranes. Some authors consider the amnion an extraembryonic ectoderm. So it's super important layers we'll see. It helps folding. It's super important with folding. So here we have epiblast cells in this little cartoon, and they're, by mitosis, first the cavity formed, a hollowing occurred here, and it filled with the amniotic fluid. And then we need to line it. And so these cells by mitosis split, and they split, and they split, and they ratcheted up this new layer of cells. And those are called amnion cells. Or collectively, this is the amnion. Got it? Another nice picture of the amnion here. Now this is implanted completely. We get a little plug forming here. Yeah, we'll talk about Heuser's membrane in the next lecture. The same kind of story is going to happen down here, where we're going to get another layer of cells. Chorionic cavity is getting really big here. Or I forgot the name of it. Must be time to stop. But this cavity is going to be lined off by Heuser's membrane, it's called. The blastocyst cavity. Okay, that must be about time to stop. And is it? Aren't we going to stop? Not yet. So, like when I made these slides, Game of Thrones was super popular. They're invading. I was all excited about this. But the syncytiotrophoblast, this is like the syncytiotrophoblast, and here's the myometrium. We're pulled going deeper and deeper, but syncytiotrophoblast cells, they divide and they increase in number, and they pull the embryo deeper and deeper, and they invade the endometrial tissue like the army of the dead. By day 10 to 12, the embryo is completely embedded in the endometrium, and then you get a little plug, a little scar that forms, which they should have drawn, a little scar that would form here. We saw that in the other one, right there. See this little scar where it went in there? Fibroblasts got in there and scarred that up. And yeah, so by day 10, 12, implantation is complete. The blastocyst is completely surrounded. It's in the endometrial stroma. It's deep to the endothelial layer. And that's where we're going to stop right there. We'll talk about hooking up the blood supply next time. All right. See you guys later.